Good afternoon, everyone, and thank you for joining my presentation today. I will be standing here because with my height I felt a bit hidden behind the booth and the laptop. So I'm Renee Moerkens and I'm a PhD student in the final phase of my PhD project. So this conference was really a nice place to get some inspiration for the next steps after my PhD. And today I will present about my PhD project, which is about steering epithelial and mesenchymal cell type composition in an iPSC-derived intestine-Chip. In our lab we study complex intestinal diseases that have a genetic component. And our main focus is celiac disease, which is an intolerance to gluten. And in order to study these intestinal diseases, we need model systems that are able to capture the complexity of the intestine. So a very short recap of a few key principles that are interesting for this presentation. So the small intestine contains these crypt villi structures. These villi are lined by an epithelial barrier that contains different subtypes. And underneath this epithelial barrier is another cell type called mesenchymal cells, and they support this epithelial barrier both physically as well as through all kind of biological signals. And the aim of my PhD project was to model this intestinal epithelial barrier. And to do so, we used the emulate intestine-Chip system. And in this presentation, I will guide you through the different steps that I've taken during my PhD to generate this intestinal epithelial barrier on chip. So, because we study genetic diseases, we want our model system to include an individual's genetic background. And that's why we make use of induced pluripotent stem cells. This also allows us to later generate different cell types with the same genetic background. So for this project, we generated induced pluripotent stem cells from the urine of 3 control individuals, and we differentiated these towards intestinal organoids. And these then contain 2 cell types, both epithelial cells as well as mesenchymal cells that co-develop. And since our main interest was really the epithelial cells, we sorted out these epithelial cells and seeded these in the top channel of the emulate intestine-Chip system. And these pictures show the cells in the system as a top view and as a cross-section. And you can see that after seeding the cells in the system and exposing them to a continuous flow through both the top and the bottom channel, they form these 3-dimensional folds that resemble to a certain extent the villi that we see in the small intestine. And these folds don't only form on a specific location in the chip, but really along the entire chip channel where the top and bottom channel overlay. And here you can see the height of the tissue in the system. So we wanted to further characterize this tissue using immunofluorescent staining. And we stained F-actin, an actin cytoskeleton marker, and ZO1, a tight junction protein. And using 2-photon microscopy, we were able to image through the tissue from the top of the tissue towards the membrane, as is depicted by this red plane. And that data is shown in this movie. So you can see in this movie that this extensive network of folds really goes all the way to the membrane. And that you can see here. And then to our surprise, we also detected cells just underneath the membrane in the bottom channel, even though we only see the cells in the top channel. And indeed, when making this cross-sectional slice, we saw that there was quite a thick layer of cells present just underneath the membrane in the bottom channel, as you can see here. So we wanted to characterize what these cells were, and we did so via immunofluorescent staining. So as expected, we saw that the epithelial cells were present in the top channel, and the cells that were present in the bottom channel were mesenchymal cells. And it might be that these mesenchymal cells still arise from the differentiation, even though we did the sorting for epithelial cells. Or there might be some kind of epithelial to mesenchymal transition ongoing in the chip. But regardless of their origin, it's very striking that consistently in every experiment these cells are present and find their way through the pores of the membrane to the bottom channel. So then we want to further optimize the intestinal epithelial composition of the system. So in the small intestine, we have these different intestinal epithelial subtypes that are organized along the crypt-villus axis, so with the pennant and the stem cells in the crypt region and the differentiated cells along the villus. And the spatial organization is largely dictated by the presence of growth factor gradients along this crypt-villus axis. And based on literature of adult stem cell-derived organoids, we composed a medium that resembles the crypt region, so really inducing proliferation. And we composed a medium that resembles the villus region, so really inducing differentiation. But if you only expose your system to villus medium, in the end you will lose your proliferative cells, which will eventually also mark the end of your experiment. So since we had a two-compartment system, we decided to introduce the crypt medium to the bottom side, to the bottom channel, and introduce the villus medium to the top channel, and hereby try to really recreate this gradient. And we then wondered whether such a gradient could give rise to a more diverse epithelial population that better reflects the human small intestine. So after seeding the cells on the system and letting them grow for a few days, we exposed them to either the crypt medium on both sides, the villus medium on both sides, or to this gradient. And this is how the cells look like under these different conditions. So we can see here in the top panel the top view of the top channel, and in the lower panel you see the cross-section of both the top and the bottom channel. And we can see that after exposing the tissue to crypt medium on both sides, you get this really dense network of folds and a high tissue. After exposing to gradient, these folds are maintained, but they are a bit less dense and the tissue is a little bit less high. And after exposing to the villus medium, the fold density is quite greatly reduced and the tissue gets quite thin. So this was nicely in line with our hypothesis of really inducing proliferation versus inducing differentiation and having this intermediate state that might still have both. So here I show some immunofluorescent images of the cross-sectional slices. You can see here in red the intestinal epithelial cells and in yellow the mesenchymal cells, as we were also interested to see how these cells would respond to these different conditions. And we can see here again that the epithelial cells form folds in the top channel in all conditions, and the mesenchymal cells are present in the bottom channel in all conditions. And after exposing to crypt medium on both sides, not only the epithelial cells largely proliferate, but also the mesenchymal cells. And you can see that in some cases, they were now even present in the top channel as well. And after exposing the system to the gradient condition, we reduce these mesenchymal cells. And after exposing to villus medium, we further reduce these mesenchymal cells, and only very few are left, together with a reduction in the epithelial tissue. So it seems that this gradient condition is a good way to preserve your epithelial cells as well as your mesenchymal cells and restrict these to the bottom side of the system. Then we also did the staining for goblet cells, which you can see here in green. And very few of these cells were present in the crypt media condition, and we can successfully induce these in the gradient and the villus media condition. And the same is true for enteroendocrine cells, or the hormone-producing cells. Few of these are present in the crypt medium, but in the gradient and villus media condition, we can successfully induce them. And here I show enterocytes, so the cells that are responsible for digestion, in yellow. A few more of these cells are already present in the crypt medium, but we can further enrich these enterocytes in the gradient and the villus medium. Then we also wanted to get a little bit more unbiased insight in the cell type composition in the system. And for this we did single cell RNA sequencing. And that data is shown here in this plot. So every dot represents a cell, and they are clustered based on similarities in their transcriptomic profile. And this plot shows data of all three conditions together to really show you the different subtypes that we find in the system. And we were happy to see that we find all the major subtypes that we also see in the human intestine back in our iPSC-derived intestine-Chip. So here on the left side you see the epithelial cells, so you can really see the proliferating cells in the middle here in the core, and the enterocytes differentiating out from these. Here the Paneth cells, goblet cells, and enteroendocrine cells. And then on the right side you see the mesenchymal cells. And keep this division in mind for the next slides, because here I separate the data based on condition. And you can really see that after exposing to the grid medium, you have this dense core of proliferating cells and very few of these differentiated cells. And we see a lot of mesenchymal cells. In the gradient condition, we have both proliferating cells as well as progenitor cells and differentiated cells. And in the villus medium, we have more an enrichment for the differentiated cells and fewer proliferating cells and mesenchymal cells. So it seems that in the gradient condition, we can really capture the differentiation trajectory that's happening along the crypt-villus axis in the human intestine. And then we wanted to compare our data of the intestine-Chip to data of the human intestine. And for this, we use data of the gut cell survey, which contains both different age groups, so from fetal to adult, as well as data of different intestinal regions. So first we predicted the age of our intestine-Chip, and you can see here that it's predicted to be fetal in phenotype, and that is actually seen more often in iPSC-derived model systems, and only the most differentiated cells on the outer edges were predicted to be a little bit older in age. And here I show the prediction of the region. So most of these cells were predicted to be small intestinal in phenotype, with a few patches of large intestine. Then we looked a bit further into the epithelial composition. So here I show the epithelial to mesenchymal ratio. So in green you see the epithelial cells relative to, in orange, the mesenchymal cells over the different conditions. And we can see what we have also seen in the microscopy images. So we can see this enrichment for epithelial cells in the gradient and villus medium compared to the crypt medium. And interestingly, when we compare this data to data of the human intestine, we see a similar trend in the developing human intestine from fetal to pediatric to adult. Also an enrichment in these epithelial cells. So even though our system or the individual cells were predicted to be fetal in phenotype, the cell type composition actually resembles more the cell type composition of the adult human small intestine if we're talking about the gradient and villus medium condition. Then I want to zoom in a bit further on the epithelial composition that is shown here, and we can focus on the major groups that we see, so proliferating cells, Paneth cells, and enterocytes. So we see a reduction in proliferating cells and an induction of enterocytes in the gradient and villus medium condition relative to the crypt medium condition. And this now resembles roughly the composition of the human small intestine, except that we still see quite a lot more Paneth cells in our system compared to the human intestine. And here I show the plots for the goblet cells and the enteroendocrine cells, so you can see this induction that we've also seen in the microscopy data, and it's now to a similar level or even to a higher level as we see in the human adult small intestine. Now that we know that we have all these different subtypes in the system, we also wanted to see whether we could find evidence that they are also functionally active. So here I show significantly different genes between the conditions for epithelial cells, and we can see genes are upregulated in the gradient and villus medium condition that are related to important intestinal functions. So you can see here functions as digestion, drug metabolism, and mucin production. While genes that are related to proliferation are upregulated in the crypt media condition. And then interestingly, we also found some genes that were related to different signaling pathways that are involved in steering intestinal cell fate towards either a stem cell or a differentiated cell. And for this, we should not only be looking at the epithelial cells, but also definitely at the mesenchymal cells. because the mesenchymal cells actually secrete a lot of the ligands that can be sensed by epithelial cells to steer them in a certain fate. So we looked into our data whether we could find evidence of this epithelial to mesenchymal communication. And here I showed two examples of potential communication. So you can see here in the right upper pane that the mesenchymal cells produce BMP4 and that the receptor is present on epithelial cells, and BMP4 is related to the induction of differentiation of the epithelial cells. And the mesenchymal cells also produce Wnt5a, which is important for the maintenance of stem cells. And the receptor is almost exclusively present on the epithelial cells. So this data suggests that it might actually be beneficial to preserve the mesenchymal cells in your system, just like we do in the gradient situation, as they might have a supportive role for the epithelial cells, just like in our own intestine. And that brings me to the take-home messages. So I've shown you that adjusting media composition allows you to steer the epithelial and mesenchymal composition in an iPSC-derived intestine-Chip, that by combining crypt and villus media, we can induce physiologically relevant epithelial, mesenchymal, and epithelial subtype compositions, but not yet spatial organization. So this is something that we're really still improving. And that by including mesenchymal cells in a model of the intestinal epithelial barrier might actually be beneficial for the epithelial cells and opened up new avenues for research, because these cells are more and more implicated in different disease phenotypes. So with that, I would like to thank you for your attention and thank all the amazing people on these slides for not only making science a lot more efficient, but also a lot more fun. Thank you.
Any questions for Renee? We have a couple of minutes. Yeah.
Hello, excellent talk. I have two quick questions. I might have missed it at the beginning, but these two different media types that you generate that gradient of, what are the key distinguishers between those two types? And then from what I understood, you were able to separate the EpCAM positive cells, and that's what you started off with in your organoid. How does culturing, like the viability of just culturing those epithelial cells look like?
With different conditions, you mean then,
Yes.
right? Yeah, okay. So the differences between these different media types are mainly that the villus medium has no activation of the Wnt pathway and has notch inhibition and MAP kinase inhibition in there. So to really steer more towards a secretory phenotype. And your second question about viability. So we see that, so we tested all this also in organoids and transwells as pilots, and we see that the viability is a bit lower in the villus medium, also depending on how long you expose the cells to this villus medium. But that especially that is related to putting in MAP kinase inhibition, which you have to titrate really carefully, and if you're at the good range, viability will actually stay the same, but you just stop the cell division and you can keep them for a few days. So the viability in intestine-Chip in the end wasn't very different for these different conditions, because we titrated the MAP kinase inhibition very carefully.
Any more questions? Yeah.
Thank you very much. It's really great data. I was wondering, you said that you might have some EMT in your epithelial cells, but that would also mean, at least to my knowledge, most of the mesenchyme in the intestines is not deriving from the endoderm, but from the mesoderm. So you might even have a transition of the germ layers. Do you know if there's any data on that from, let's say, adult intestine stem cells? Because those would then maybe also do that transition?
So from adult intestine, well, there is EMT ongoing, mainly in the context of disease, as far as I'm aware. But you're totally right that within the differentiation of an iPSC to intestinal stem cell, you also have a little bit of mesoderm there. So that is one origin definitely of the mesenchymal cells being there. But in the both the FACS data and the single cell data, I see some indications that there is an epithelial or an EpCAM and Vimentin double positive population. So I think it probably goes both ways. So still some cells from the differentiation and also some EMT in the CRIP medium where we don't inhibit MEP kinase.